This video here is going to be specifically about prokaryotic cell walls. And the most important thing that you will learn in this section here is that you're going to be able to compare and contrast the cell walls of gram-positive bacteria, gram-negative bacteria. That's going to be the most important part right here. Gram-positive versus gram-negative bacteria and how their cell walls are different. We also have some exceptions to the regular gram-positive, gram-negative cell walls, and that will be some exceptions to the acid-fast bacteria. We're going to take a brief look at the archaea cell walls, and then mycoplasmas, which don't have a cell wall. And that's what I want you to get out of this section right here. So we're going to focus on prokaryotic cell wall, specifically the gram-positive versus the gram-negative cell wall. So the cell wall is great because it protects the cell from lysis, for example. So let's say you have a freshwater organism, and normally it would swell up and burst if it didn't have a cell wall, but the cell wall protects it from lysis, osmotic lysis, and also it's just a real nice layer of protection. In prokaryotic cells and regular bacteria, the eubacteria, the cell wall is made out of something called peptidoglycan. Okay, that's a very important term that you need to know. And the peptidoglycan, just like the name indicates, peptido refers to protein, glycan refers to carbohydrate. So it is a mixture of protein and carbohydrate. And we should take a look at that. So here first I have a nice electron micrograph that shows you the cell wall. And then this bacterium has a capsule on top of that, but the cell wall is right here. So that's this part right here. Sort of in purple, I don't know if you can see that, but the cell wall is underneath, and then the capsule is on the outside. That's the outer layer of that. And here again, the cell wall, let me change the color of my pen. So the cell wall again is underneath the capsule, so it will be right here. It's a bit of red right there. It goes all the way around, of course. So that's the cell wall. And then the capsule is on top of that. So the capsule is on the outside. That's the fuzzy stuff, fuzzy looking stuff that's right there. So that's in this electron micrograph. Now, let's take a look at the composition and the characteristics of the prokaryotic cell wall. And again, I'm focusing here strictly on the eubacteria. The archaea are completely different. But for eubacteria, we have this peptidoglycan cell wall, and it is a polymer of repeating disaccharides. In the previous lecture, we learned what monosaccharides and disaccharides are. So these are carbohydrates. Monosaccharides are simple building blocks, and if you put two of them together, then you're going to have a disaccharide. So we have repeating rows of a disaccharide, and these two, the disaccharides in question here are NAG and NAM. And NAG stands for N-acetylglucosamine, that's right here, and then we have NAM is the N-acetylmuramic acid. NAG and NAM alternating, forming the carbohydrate component of the bacterial cell wall. And then these rows of carbohydrates, they are linked by polypeptides, which are composed, of course, of amino acids. So that would be a protein type of chemical. So here is the NAG and NAM component. So these are disaccharides. So we have N-acetylglucosamine, NAG, and NAM, N-acetylmuramic acid, linked by a glycosidic bond. So this would be a disaccharide. And here's what it looks like. So we have our NAG and NAM is here color coded in pink and brown, I guess. So NAG and NAM are here. NAG, NAM, NAG, NAM. So you have these rows of disaccharides. Rows of disaccharides going down this way. Okay, and now these sort of tubes that you see right there, these alternating, or these disaccharides that are linked up together, these sort of NAG, NAM disaccharides. These guys need to be cross-linked and need to be held together, and there you can see right here, these are the cross-linking polypeptides right here. So these little spheres that are, you can see right here, that are cross-linking the whole thing, that is the protein component. So these amino acids, they are linked by peptide bonds, and we learned that in the previous lecture. So this is what it looks like. So now let's take a look at the gram-positive versus gram-negative cell wall. The gram-positive cell wall has thick peptidoglycan. So gram-positive cell wall, thick peptidoglycan, versus the gram-negative cell wall has thin peptidoglycan. Okay. And that makes a big difference when you are staining these bacteria, because the thin peptidoglycan of the gram-negative cell wall will not be able to hold back the washing out of the crystal violet in the gram staining procedure. So once you are washing your cells with alcohol, then the crystal violet will wash out because the peptidoglycan layer is too thin in the gram-negative bacteria. Also, we have teichoic acid that we find in gram-positive cell walls. And in the gram-negative cell walls, we have an outer membrane that's composed of phospholipids. And we do have a periplasmic space that is not present in the gram-positive cell wall because we just have the two layers. Okay, so let's take a closer look at the gram-positive cell wall only. So we have teichoic acids, and basically these wall teichoic acid, they link the peptidoglycan. So this teichoic acid links the peptidoglycan, carries a negative charge, and it regulates the movement of cations. And so that gives additional properties to the gram-positive cell wall. The polysaccharides and teichoic acid, they provide antigenic specificity. That means they give they're like little red flags for your immune system. So when we talk about antigenic specificity, the antigenic properties refer to how your immune system deals with these polysaccharides and the teichoic acid. So they're species specific to the different bacteria, and how your immune system reacts to them determines how they make you sick, basically, and how much damage they can do to you. Okay, so let's take a look here at a diagram of the gram-positive cell wall. So we have our layer of phospholipids, that's the plasma membrane down here. So here this would be just the inner phospholipid bilayer. And then we have in tan and pink, I guess, this would be our NAG-NAM grid right here. So these tubes, that's still the disaccharides. These they're sort of arranged, or parallel arrangement. So the tubes, that would be your carbohydrate component of the peptidoglycan. And then here in green and blue, that's the protein component of the peptidoglycan. So in green and blue, let me circle this on another one. These are your cross-linking peptides that are holding the carbohydrate NAG-NAM structures together. And in the gram-positive cell wall, you also have the wall teichoic acid. These are these black lines that are sort of stabilizing everything. Okay, so this is the gram-positive cell wall. Just one phospholipid bilayer, and then very thick peptidoglycan, so a really thick layer of these alternating carbohydrates cross-linked with peptides and stabilized with teichoic acid. So that's gram-positive. And now we're going to take a look at the gram-negative cell wall. There we have this periplasm between the outer membrane and the plasma membrane. It contains a thin layer of peptidoglycan, and then the outer membrane is made of polysaccharides and lipoproteins and phospholipids mostly. And we should just take a look at that. So the gram-negative cell walls are a little bit harder for your immune system to scout out, because phospholipids, that's what human cells have too, and so they're kind of they don't raise flags for your immune system. And so gram-negative cells in general are a little bit harder to treat, and they are harder for your immune system to see. So these gram-negative cell walls in general, they protect from phagocytes, like your macrophages and neutrophils, also they don't allow for complement activation, and it makes it much harder to treat with antibiotics as well. Although there are some antibiotics that are good for use with gram-negative bacteria. But anyway, so we have a specific to gram-negative cell walls are these lipopolysaccharides, LPS. And you already heard from that particular strain of E. coli that can make you sick. There's the O157H7 strain. We heard already about the flagella component of that's the H7 right here. But then you also have the polysaccharide, the O antigen. So the O157, this one here refers to the cell wall polysaccharide component. So again, the O polysaccharide actually functions as an antigen, so that's a component of the gram-negative cell wall. That's something that does cause an immune reaction. And here the example is given, the O157, so this is that O polysaccharide. This particular one causes a pretty bad immune reaction. And now again, the H7, that's the flagella antigen, and it also causes an immune reaction. And then we have lipid A, that's an endotoxin and can be embedded in the top layer of the gram-negative cell wall, and that creates another problem. Sometimes these endotoxins they can cause high fevers, definitely cause a reaction, and they are only released from the gram-negative cell walls after a phagocytic cell has eaten those gram-negative bacteria and has then digested them, and in the process of digestion it's actually releasing this endotoxin that then can cause rashes and fever and other bad things. Anyway, another thing about the gram-negative cell wall are these porins. These are protein channels that go through the membrane. Now let's take a look at this. So here is your gram-negative cell wall. So to me, it reminds me to an American sandwich. Bread on the bottom, bread on top, and stuff in between. That's how Americans like to eat a sandwich. Believe it or not, this is not the only way you can have a sandwich. I'm from Europe originally, and in Europe, we never have bread on top when we eat a sandwich. Bread to me was always just bread and then stuff on top, like the gram-positive cell wall, where you just have bread on the bottom and then you put your stuff on top and then you eat it with a knife and fork. In the United States, most people will make a sandwich. To them, it's a sandwich when you have bread on the bottom, bread on top, and then your stuff, sausage and whatever, cold cuts and your vegetables, you have that in between. So anyway, you can maybe, I don't know, picture that if you like, but the gram-negative cell wall resembles more an American sandwich. We have a phospholipid bilayer with, of course, proteins and stuff and embedded in there, but here's your phospholipid bilayer on the bottom. Then we have a phospholipid bilayer on top that has here some porin proteins are embedded into that, but nevertheless it's a phospholipid bilayer. Okay, so it also contains this O polysaccharide and then lipid A and whatnot. And then in between, so we have an outer membrane and an inner membrane. And then in between these two membranes, we're going to have our peptidoglycan. And you can tell you have only one row of nags and nams that are then cross-linked by some peptides. So the peptidoglycan is pretty thin, significantly thinner than the in the gram-positive cell wall. And sort of the space between the two phospholipid bilayers, we call that the periplasm. So that's something for you to consider. Or maybe one more thing here, there the O polysaccharide that we talked about earlier that can cause an immune reaction. That's sort of what these blue things that are sticking up. And that O157 in that particular type of E. coli causes a pretty bad immune reaction. Okay, so let's talk about the gram stain. Of course, it's very important for microbiology students. Here are the steps. And so because these two different types of cell walls give the bacteria quite different properties, and also they're forming two groups when it comes to how you want to treat a bacterial infection. You have to really have a different approach between gram-positive and gram-negative bacterial infections. But here is the gram stain. That's very important because it distinguishes these bacteria based on their cell wall properties. And the first thing in the gram stain, the first step is that you, after of course you smeared your bacteria, you fixed them, and now they are stuck on your slide. The first thing you want to do is you want to add crystal violet. So the crystal violet is a deep purple dye, and we're gonna try that out and do that as well. So you have some crystal violets in your kits. The crystal violet will enter the cells and will make all the cells that are on your slide purple, no matter whether they're gram-positive or gram-negative. The crystal violet will go inside. Next step will be that you're gonna add iodine as a mordant. The grams iodine, it's a liquid, but when it comes in contact with the crystal violet, it forms sort of a thicker or larger complex. And then when we are going to try to decolorize these cells, then the crystal violet iodine complex will be too thick, and because the gram-positive cells, they have this very thick peptidoglycan layer, so the crystal violet iodine combination is retained. So in the gram-positive cells, when we try to wash out the crystal violet iodine complex, then two things will happen. The alcohol, first of all, it dehydrates the peptidoglycan and makes it even more impermeable. It just makes it a real kind of a thick layer that's just, there's not, the dye will not be able to leave. And so the crystal violet iodine complex will be retained and it will not be able to leave. Okay, so the alcohol wash, that's the distinguishing step. The thick peptidoglycan of gram-positives will be even made thicker by the alcohol, or maybe even more dehydrated and tougher, I should say, and the crystal violet iodine will not be able to leave. Now, different here for the gram-negatives. The gram-negatives, they have an outer phospholipid bilayer, and the alcohol ends up dissolving that outer membrane, because you might know alcohol and lipid, it's sort of a way to clean up or to dissolve the lipid. And so now we only have a very thin layer of peptidoglycan, and it will not be able to retain the crystal violet iodine combination. So the CVI, the crystal violet iodine, washes out, and the cells have no more color. So now you would have on your slide, you would have purple gram-positive bacteria, but you wouldn't be able to see the gram-negatives because they have lost all color. And so we need to have contrast. So you need to add safranin as your last step, and it's a kind of a pinkish dye, and that gives the contrast back to your gram-negative cells. So this is the gram stain, and you need to know it. So there's no way around it. All microbiology students need to know the steps of the gram stain and how it works. And if you did everything right, then you should see something like this. Gram-positive cells will show up dark purple from the crystal violet that they retained. You see that over here. That was the gram-positives. Gram-negatives, they will have lost the crystal violet, so they're no longer purple, and they will retain this pinkish red kind of dye from the safranin. Safranin is like a reddish, pinkish red kind of dye. Okay, so that's what it should look like. And here, sort of a summary. Gram-positive cell walls, they attach flagella differently. So they have two rings in the basal body to attach to flagella. They produce exotoxins, that's important. They have high susceptibility to penicillin. So gram-positives are easily treatable with penicillin. And their cell walls are disrupted by an enzyme known as lysozyme. Gram-negative cell walls, they require 4 rings in the basal body of the flagella to attach a flagellum to that gram-negative cell wall. They produce endotoxins a lot of times. So these endotoxins are embedded in the gram-negative cell wall, and the gram-positive cells don't have that. And these endotoxins are oftentimes released when these cells are being digested by phagocytic cells. They also can have exotoxins in addition to the endotoxins. And gram-negatives show low susceptibility to penicillin. Okay, so now let's take a look at atypical cell walls to finish this section off. We have acid-fast cell walls. They're atypical. They're even harder to treat than, these are some of the toughest bacteria that we know. And they are in general like gram-positive cell walls, but even tougher. They're adding another component like microlic acid, in addition to all the other components that the gram-positive cell walls have. So the mycolic acid makes these cell walls even tougher. And the mycolic acid is found in Mycobacterium, tuberculosis, for example, Mycobacterium leprae, all of the mycobacteria. And then also the Nocardia species of bacteria. You can stain them with carbofuscin. And here is a stain. Carbofuscin is a reddish stain. You can see acid-fast bacteria, and they retain the dye even if you wash these cells with acid alcohol. Therefore, we call them acid-fast. And so normally, even gram-positive cell walls, if you hit them with acid and alcohol, they will lose their dyes. But if you have mycolic acid in addition to the gram-positive cell wall components, then they will retain the dye, and those are really tough cell walls. So the mycobacterium right here, tuberculosis, the staining, the acid-fast staining here, the pink bacteria, that's what we're looking for. Stained with carbofuscin in this case. More atypical cell walls, the mycoplasmas, they don't have cell walls at all, so they lack cell walls, but they have sterols in the plasma membrane. And then the archaea are completely different. They either don't have a wall at all, or they have something called pseudomurine. So they don't lack, they have the NAM and the D amino acids. They don't have that. Those are components we find in the eubacteria, but in the archaea, the walls contain pseudomurine, so it's a different kind of thing. Or they don't have a wall at all. Okay, damage to the cell wall. A lot of antibiotics try to attack the cell wall, and it works specifically well with the gram-positives, but we'll get to that. We have a whole lecture on how penicillin works. So lysozyme is an enzyme that we have also in our mouth. It hydrolyzes the bonds in the peptidoglycan, and so therefore keeps your bacterial population in your mouth sort of in check. But lysozyme in general, it hydrolyzes the bond in the peptidoglycan, and so therefore it basically ruins the cell walls of eubacteria. Penicillins, they will inhibit peptide bridges in the peptidoglycan, and so therefore you're making very weak cell walls, and these cells, they cannot withstand lysis by osmosis, and so they die very easily. And another thing is protoplast, another term you should know. These are gram-positive cells that have lost their cell wall. In general, we call something a protoplast if you have digested away the cell wall, and so now the only thing that's left is a plasma membrane. So they're very fragile. They've lost their cell walls. And then something not that important, spheroplast, the wall-less gram-negative cells, and then L forms, also not that important for you guys, wall-less cells that swell into irregular shapes. But this is it. This is all I have on cell walls. I'm going to stop the recording right here. Make sure that you know this section. You can check your understanding if you like. As I said, we don't really pay attention to the L forms, but you should know that mycoplasmas, they don't have cell walls at all. And we will spend quite a bit discussing how drugs like penicillin target cell wall synthesis. Okay, so make sure that you know the gram stain every step, and make sure that you can distinguish and describe the differences between gram-positive and gram-negative cell walls.